Hello, Biology 202 students. Welcome to your online lecture. I'm excited to guide you through today's fascinating topic. It's blood. This lecture will provide you with a comprehensive understanding of blood's components, functions, and its critical role in the human body. We'll explore the historical and cultural significance of blood, its modern medical applications, and delve into the classification of a connective tissue. By the end of this session, you will have a solid grasp of how blood supports various physiological processes and maintains overall health. Let's get started. In today's agenda, we'll be talking about the historical and cultural significance, blood as a connective tissue, the components of blood, which are formed elements, red and white blood cells with plasma, and then we'll talk about blood typing. Blood has been central to many religious, spiritual, and mystical traditions. Historically, blood was sprinkled on walls and statues to honor deities and used to thwart evil spirits. Personally, I would start with burning some sage, but to each their own. Stories of mystical beings such as vampires who rely on drinking blood for sustenance run rampant in many cultures, including pop culture. When blood is left to settle in a container, it separates into distinct layers. This phenomenon led to the ancient Greek and Roman medical theory of humorism, which proposed that the body was governed by four humors: black bile, yellow bile, phlegm, and blood. Health was thought to result from their balance and disease from their imbalance. This theory persists until the mid-1800s, when it was debunked by Rudolf Virchow. Virchow is known as the father of pathology, and he is worth reading about. Today, blood is used in numerous vital applications. It saves lives through donation, aids in archaeological studies, solves crimes, and even used in artwork. For example, contemporary artist Mark Quinn created a self-sculpture using 10 pints of his own blood. It may be surprising, but blood is actually a connective tissue. Blood has several crucial functions. It transports oxygen from the lungs to tissues, delivers nutrients absorbed from the digestive system to cells, removes waste products for excretion, and distributes hormones produced by the endocrine system. As a connective tissue, blood supports and connects many tissues in the body, playing a vital role in maintaining overall physiological balance and health. This is known as homeostasis. Blood consists of two major components, formed elements and plasma. After centrifuging blood, it will separate into three distinct layers. On the top, there'll be plasma, then just below that, the buffy coat, and underneath that, erythrocytes. And so you can see plasma makes up about 55% of total blood volume. And the buffy coat, which has leukocytes and platelets, that makes up less than 1% of total blood volume. And erythrocytes, those make up 45% of total blood volume. So let's talk about erythrocytes. Erythrocytes are the red blood cells. They are the most common blood type, or blood cell type, and they make up about 5 million per cubic millimeter of blood. Each erythrocyte contains approximately 250 million hemoglobin molecules, enabling the transport of about 1 billion oxygen molecules per cell. Next, there's leukocytes. Leukocytes are white blood cells. They are key players in the immune system, and they're formed in the bone marrow. And they're capable of living from a few hours to several years. They're divided into granular and agranular groups. So granular cells are the neutrophils, the eosinophils, and the basophils. And the agranular cells are lymphocytes and monocytes. The biggest difference between these two groups are their visual appearance. The agranular, they don't have visible granules in the cytoplasm, while the granular do. And you'll see that in some later images. Next, we have thrombocytes, or platelets. They are small cell fragments involved in clot formation, and they originate from megakaryocytes in the bone marrow. Lastly, we have the plasma. The plasma is mostly made of water, so it's like 90% water. And the other things in the plasma are proteins like albumins and globulins and fibrinogens, but there's also electrolytes, some nutrients, and hormones. Remember from last lecture? Yeah, there are hormones in the blood, and it's actually in the plasma. And there's also waste in the plasma, and we'll talk about that in later lectures as well. This diagram illustrates the components of blood. So first we have the whole blood, right? It hasn't been separated into its formed elements and plasma. But we can see what consists of the plasma. All of this stuff, you can pause and review that. And then really what I want to draw your attention to are the leukocytes and how they divide into two groups. We have granular and agranular. The granular are the neutrophils, eosinophils, and basophils. The agranular consist of monocytes, lymphocytes, okay? But lymphocytes can further subdivide. So lymphocytes actually encompass B cells, T cells, and natural killer cells. Here is a histology slide of whole blood. So you can see all the elements. We have the formed elements all here, and presumably there would be plasma between these cells if it was in the body. So there are a ton of these light pink circles. And since these are the most popular cells, those would be erythrocytes. There's also thrombocytes out here. They're just these tiny specks in the histology slide. And then leukocytes are all the ones that stain darker purple, and we will discuss those later. Something you might notice with this image of red blood cells is some of them actually have a clear center or a little white spot. And that's because that's the thinnest part of the cell. So erythrocytes are actually biconcave, which means they have a large bulbous outer rim and a thinner interior. That thinner interior is because there used to be a nucleus there. But the red blood cells don't need nuclei. They need to be able to hold oxygen. So let's dive deeper into the fascinating structure of these cells. Erythrocytes, their main function is to move gases from tissues, right? We move oxygen to our tissues to give them what they need for metabolism, and then we take away the waste, which includes carbon dioxide. As they're developing, they actually get rid of their nucleus to maximize their gas exchange. Because of this shape, they actually have a better surface area to volume ratio. So it's about 20-30% greater than if it was just a spherical cell. So the shape actually does help a lot with gas exchange. Now, when we look at the shape, we're also looking at the membrane. So the membrane of the erythrocyte is a phospholipid bilayer, like most other cells. There are these proteins which provide the cells with structural integrity and flexibility, allowing them to change shape as they travel through the bloodstream. So some vasculature is actually quite small. Capillaries are very small, and they can actually be smaller than the red blood cells. And so the red blood cells will actually change shape so they can fit. They're like fold a little bit to fit in there. Some other interesting things about red blood cells are their lifespan. So each red blood cell has a lifespan of about 120 days. During this time, they travel about 250 miles through your bloodstream. They deliver their oxygen to the tissues, and they pick up the carbon dioxide for elimination. In their 120-day lifespan, red blood cells complete about 170,000 circulations around the body. The human body produces about 2.4 million new red blood cells per second. 1/1000. All right, that's 2.4 million red blood cells that have been produced to replace the ones that have aged out. And as those red blood cells age out, they are broken down by the spleen and liver, where their components are then recycled through the body. Now let's talk about the erythrocyte pathology, which involves the study of diseases and disorders affiliated with red blood cells. Understanding these pathologies is crucial for diagnosing and treating various conditions. So here we have some key pathologies. Let's start off with anemia. So anemia is a condition characterized by a decreased number of red blood cells or hemoglobin, leading to reduced oxygen transport. There's a few different kinds, but we'll talk about iron deficiency anemia because it's simpler to understand. So really, because there's a lack of iron, there is less hemoglobin to transport oxygen throughout the body. And some symptoms that encompass the anemia is fatigue, weakness, pale skin, shortness of breath. And so you can see there's a difference here in the erythrocytes. Here are some normal ones, and here are some affected by iron deficiency anemia. You can appreciate that the white centers of these cells looks like it's spreading, and that's because the thin portion of the cell is spreading out. The whole cell is getting more thin. Next, there is the sickle cell disease. So sickle cell disease is a genetic disorder resulting in abnormally shaped erythrocytes, which can block blood flow and cause pain. So it's actually quite painful to have sickle cell disease. So the pathology is actually the mutated hemoglobin. The hemoglobin causes the erythrocyte to become rigid and a crescent shape when it has mutated. Lastly, let's talk about malaria. Malaria is caused by a parasite, which is transmitted to humans through a bite from a mosquito. So once inside the human, these parasites target and infect erythrocytes. So here there's a special dye used so you can actually see these parasites. Now, fun fact, did you know that conditions like sickle cell disease can provide a survival advantage in malaria endemic regions? The presence of sickle cell hemoglobin provides some resistance to malaria, and this is something interesting that highlights an evolutionary trade-off. Now let's shift our focus to platelets or thrombocytes, which play a crucial role in blood clotting and hemostasis. Platelets are the smallest blood cells, typically around 2 micrometers in diameter, and they don't have a nucleus. They have an average lifespan of 7 to 10 days. There are about 150,000 to 350,000 platelets per cubic millimeter of blood. They were first recognized by their role in hemostasis over 100 years ago by the Italian pathologist Giulio Bizzozero. Significant advancements in our understanding of platelets have been made using techniques like electron microscopes and immunofluorescence. But this is what Giulio Bizzozero saw. We have these pink cells. Remember what those are called? Erythrocytes. And then here, these little spots. Those are the platelets or thrombocytes. So here we have an electron microscope image of platelets. So these are very highly magnified. This one here is 30,000 times magnified. Okay, so we're looking very small. And you can see there's folds in this. So this is a platelet that has not been activated. But you can see as it is activated, you get these tendrils reaching out to form a fully activated and spread out platelet. Platelets are formed by the fragmentation of megakaryocytes in the bone marrow. These giant cells produce platelets by duplicating without dividing, resulting in cytoplasmic fragments that become individual platelets, which are then released into the bloodstream. There are many pathologies associated with platelets. If a clot makes its way to the heart, lungs, or brain, it can cause serious damage because it can actually block off blood flow. So when a clot forms, it's called an embolism. So you can get pulmonary embolisms, which block blood supply to the lungs, which can kill lung tissue. You can have a blood clot go to the heart and coronary arteries, where it can cause a heart attack or a myocardial infarction. And then it can also go to the brain, which can cause strokes. Leukocytes play an essential role in both innate and adaptive immunity, so let's talk about these. The innate immune system is the defense system you're born with. It consists of barriers like the skin, actions like coughing, the tears that protect your eyes, and the mucus on all of our mucous membranes. It provides nonspecific defense mechanisms that activate immediately or within hours of an antigen's appearance, reacting to microorganisms or foreign substances without prior exposure. Innate responses happen automatically on first contact. This system is ancient and highly conserved across many species, including plants and even single-cell organisms. The adaptive immune system requires an initial exposure before the body builds up a defense. Leukocytes develop specific receptors and antibodies for specific antigens. For example, vaccines expose a person to a small amount of a virus, usually a dead virus, so that the body can build antibodies to fight the invaders in the future without causing infection. Here is another example of the innate and adaptive immunity. On the left, you can see the innate immunity and some of the cells in that circle. You can also see adaptive immunity on the right side. These are all cells that we will discuss either in lab or in the larger lectures. Right, so now let's look at these two groups again, granular and agranular. We have neutrophils, eosinophils, and basophils within the granular group, and each one of these has granules that stain. Now, neutrophils stain using a neutral dye. So that's where they actually got their name. Neutrophil loves neutral stains. Eosinophil, well, it really likes the stain eosin. Eosin is an acidic stain, and it's really red. Basophils love basic dyes, and the basic dyes that we use are darker purple. So the way you can look at these is, well, if you can't really see a lot of the granules, but you see a lot of lobes in the nucleus, we'll talk about that in the next slide, then that's a neutrophil. Eosinophils are very red, basophils are very purple, and it's really hard to see the nucleus in there. Agranular cells are lymphocytes and monocytes, and there's two cell types within lymphocytes, the B cells and the T cells. The B cells produce antibodies and the T cells destroy infected cells. Monocytes, they can differentiate into macrophages and dendritic cells, which are key for phagocytizing pathogens and presenting antigens to T cells. Let's focus on neutrophils, the most abundant type of white blood cells and key players in our body's defense against bacterial infections. Under the microscope, neutrophils may appear to have multiple nuclei. However, it's actually one nucleus that is lobed. There can be 2 to 5 lobes in a mature cell, with each lobe connected by a thin isthmus of the nucleus. These lobes can be formed in a horseshoe shape with tight constrictions or just kind of be floating around, like this picture here. The lobe's appearance helps distinguish neutrophils from monocytes, which have a single kidney-shaped nucleus. We'll see one of those in the next couple of slides. The granules within the neutrophils are actually loaded with substances that combat infections. There is lysozyme, an enzyme that breaks down bacterial cell walls. There's also oxidants, such as hydrogen peroxide, which are toxic to bacteria. There's also defensins, proteins that puncture bacterial and fungal membranes, causing the cell contents to leak out. I really recommend looking at a video of a neutrophil chasing bacteria. It really demonstrates the incredible motility and efficiency of these cells in targeting and eliminating pathogens. So remember, neutrophils are motile and phagocytic, meaning they destroy, they eat these microbes. Next, we'll talk about eosinophils. These are a little more rare, but eosinophils have a bilobed nucleus. Occasionally, they might have 3 lobes, connected by a thin strand. Their cytoplasm's color closely matches that of red blood cells, which can make them harder to find in a microscope. However, once identified, they are very distinct due to their large, coarse granules that stain bright red or orange due to the eosin dye. The granules within the eosinophils actually contain stuff as well. So they have antihistamine molecules. These counteract histamines. They reduce inflammation caused by basophils and mast cells. There's also toxic molecules within those granules. These are effective against parasitic worms, which can enter the body through the skin or by consuming raw or uncooked fish or meat. Eosinophils are also capable of phagocytosis, especially effective when antibodies bind to the targets, forming an antigen-antibody complex. The life cycle of eosinophils closely mimic that of neutrophils. They're born and mature in the bone marrow. They circulate through the blood, and then they migrate to tissues, particularly those exposed to the external environment, such as the skin, lungs, and the GI tract. So, the function of eosinophils, they are primarily involved in combating parasitic infections and mediating allergic reactions. So, some clinical significance. If we have high eosinophil counts, that can be an indication of allergies, parasitic worm infections, and some autoimmune diseases. For instance, an elevated eosinophil count is common in patients with asthma and during allergy season. Low eosinophil counts are rarely clinically significant but can occur due to drug toxicity and stress. Our last granular leukocyte is basophils. They are the least common type of white blood cells, yet vital for our body's inflammatory and allergic responses. Under the microscope, basophils have a bilobed nucleus, but it's really hard to see because of their large, coarse granules that stain dark blue or purple with basic dyes. These granules are so numerous that they can hide the nucleus, making basophils distinct under the microscope. So the granules, they contain histamine, which can cause vasodilation and increase blood vessel permeability, contributing to inflammation. There's also heparin, which acts as an anticoagulant, preventing blood clotting. So the functions of basophils, these granules release substances that intensify the inflammatory response. Basophils are produced in the bone marrow, and they circulate the blood briefly before migrating to tissues. Basophils play a key role in inflammatory and allergic responses by releasing histamine and heparin. High basophil counts often associate with allergies. Next are lymphocytes. Lymphocytes have a nucleus that is not lobed, but may be flattened or dented on one side. The nucleus almost fills the entire cell, leaving a thin, clear blue cytoplasm around the edge. These cells make up 25 to 33% of white blood cells. They subdivide into two groups, B cells and T cells. B cells mature in the liver and spleen in fetuses and later in the bone marrow of adults. They become plasma cells and produce antibodies, contributing to antibody-mediated immunity. T cells are involved in cell-mediated immunity as well. They target infected or cancerous cells. You don't need to be able to differentiate these two, and you'll learn more in the larger lectures. The real thing you need to know is how to identify them and know their role. Lymphocytes increase in number during viral infections, such as mononucleosis, and during antibody-antigen reactions. They are key players in the adaptive immunity, developing specific responses to pathogens. Monocytes are large cells with a kidney bean or horseshoe-shaped nucleus that lacks tight constrictions. This, along with their large size, helps distinguish them from neutrophils, which may have lobed nuclei, but with tight constrictions. Monocytes make up 3 to 8% of white blood cells. Monocytes are activated by signals from T cells. They are major phagocytes, engulfing foreign materials and pathogens. When monocytes leave the bloodstream and enter tissue, they become macrophages. Macrophages phagocytize debris, foreign pathogens, and worn out erythrocytes. They also release antimicrobial defensins and chemotactic chemicals that attract other leukocytes to infection sites. Some macrophages remain fixed in tissue while others wander through the tissue fluid. High monocyte counts are seen in viral or fungal infections, tuberculosis, some forms of leukemia, and other chronic infections or diseases. Low monocyte counts typically result from bone marrow suppression. We're going to cover infectious mononucleosis. It's going to be our leukocyte pathology. Infectious mononucleosis, often referred to mono, which is caused by the Epstein-Barr virus. Mono is a common virus that most people are exposed to at some point in their lives. However, only a few people develop mononucleosis after exposure. The virus initially infects the pharynx, leading to a sore throat. It then infects B cells, where it proliferates. T cells, particularly natural killer cells, attack the infected B cells to control their proliferation. This immune response results in the presence of atypical lymphocytes in the blood. When examining blood smears, not all lymphocytes will appear abnormal. You'll need to look around to find the atypical cells, characteristic of mononucleosis. If you look at this image to the right, you'll see two leukocytes. Can you tell the difference between the one on the left and the one on the right? I like to call this the splat. The one on the right, it looks like it's been thrown up against the wall and it kind of exploded. That's the one that's infected with mononucleosis. Let's do some practice. Pause the video with each of these images and try to guess what cell it is. This is a lymphocyte. See the clear blue cytoplasm on that one left side of the cell? That nucleus is a little flattened and dented. All right, next. This is a basophil. So remember, basophils are stained very dark, or dark blue or purple. Sometimes you can't see the nucleus at all. How about this image? These two cells are neutrophils. How can you guess? Well, there's multiple lobes. How about this image? This is a monocyte. There is a large kidney bean-shaped or horseshoe-shaped nucleus there. So that's a monocyte. All right, we are in our last section for this class. This is blood typing. We have a few key terms that you need to know to be able to understand this section well. This will also help you understand some of the immune system stuff. Let's start off with an antibody. An antibody is a Y-shaped protein produced by B lymphocytes in response to an antigenic stimulus. It binds to antigens and marks them for destruction. You can see in the image on the right, the antibody is that Y-shaped protein. An antigen is a large molecule, like a glycoprotein, on the outer membrane of cells. When foreign, it generates an immune response. So you can see the antigen is that whole thing. And then on the surface of the antigen are the epitopes. And those are the pieces that the antigens actually match up with. Agglutination is the clumping of cells or molecules due to an antigen-antibody reaction. It's important to distinguish this from coagulation, which is the clotting of blood and other fluids. It's a vital process to stop bleeding. Agglutination happens when particles are tied together by antibodies. You can see in this diagram the 4 different blood groups. Now this is only half of the image because we also have Rh factor, which gives the positive and the negative. And we'll talk about that in the next slide. But here you can see each blood group and the antigens on the surface of each blood cell. These antigens tell our bodies if we have foreign or just our own blood. Our body only wants our own blood inside of itself. With red blood cells in group A, there are only A antigens on the surface. With B, there are B antigens. With AB, there is A and B. And with O, there are no antigens. So group O has no antigens. I almost think of the O as a 0 for antigens. Now, the antibodies in the plasma of these individuals are the direct opposite. If they have A antigens, they have B antibodies. If they have A antibodies, they have B antigens. If they have both antigens, they have no antibodies. And if they have no antigens, then they have both antibodies. And so what happens when a person with blood type A receives blood from blood type B? Well, the anti-B will attach and cause an agglutination between the donated blood cells. What about if they received blood from AB? Well, AB also has B antigens, so there would be an agglutination. What about O? O has no antigens, so there would be no reaction. So group A blood can receive blood only from O or another person with group A. The same idea is with B. B cannot receive blood from A or AB, but it can with O. AB blood can actually receive blood from everyone, and that's because they don't have any antibodies in their plasma. Group O can actually donate to everyone, but can't receive from anyone other than O groups. Lastly, you can see on the table to the right the genotypes associated with each one of these blood types. We have group A blood with either the genotype AA or AO, and that's because O is the recessive gene, while A is a dominant gene. So you can have heterozygous A or homozygous A. B blood is also dominant. So the B allele is dominant while the O is still recessive. So you can get AA, AO for A, and for B type blood, you get BB or BO. And then here's where things change up. You can also have A and B. They're heterozygous, but they're co-dominant. That means each one of these are dominant, and they will show the phenotype of having both A and B antigens, while the person with O type blood has to be homozygous recessive because the O genotype is recessive. Finally, let's cover the Rh factor. The discovery of Rh factor was made with rhesus monkeys. That's where the Rh part comes from. Rhesus starts with the letters Rh. Rh positive individuals have the homozygous dominant D genotype or the heterozygous D genotype on their blood cells. Rh positive individuals have an Rh factor on the surface of their red blood cells, while the individuals who have the heterozygous recessive D alleles do not have the Rh factor, so they are negative. So that's where positive and negative comes from. A positive individual has the Rh factor on their blood cells, while a negative does not. Now, this makes donation interesting because a person with positive blood can receive positive and negative, while an individual with negative blood cannot. They can only receive negative blood. An interesting pathology can arise with a mother who is Rh negative has an Rh positive baby. After the first Rh positive pregnancy, the mother's body may develop antibodies against Rh antigens, which can affect subsequent Rh positive pregnancies. This condition is preventable with medication that suppresses the mother's immune reaction. When the mother creates these antibodies, the antibodies can attack the fetus's blood cells, causing hemolytic disease of the newborn, which can cause many issues. Congratulations, you made it to the end of the lecture. I hope you have a great rest of your day.